So welcome everyone again for the CIC Lecture Weekly. I think this is session number 4. And today I'm very happy to be introducing a speaker, student in an old lab I was in. So very nice to see him again. Let me give you a bit more information. So Jonathan is a researcher working with a variety of neuroimaging modalities, including MEG and PET, to study the brain and its pathologies, notably focusing on Alzheimer's and its progression from early to late stages. Previously, he completed a bachelor's in psychology at the National Autonomous University of Mexico and a master's in neurobiology and neuroscience at the same university. Most recently, he completed a Ph.D. in neuroscience at McGill University under the supervision of Dr. Sylvain Baillet and Sylvia Villeneuve. His research focused on studying brain dynamics related to pathological accumulation of amyloid and tau proteinopathies,
yes.
in people at risk of developing Alzheimer's disease. So without further ado, I'll let Jonathan speak for himself.
Thanks for the introduction, Domenico, and thanks everyone for being here. As Domenico mentioned, I'm going to be talking about the PhD work that I conducted at Sylvain Baillet and Sylvia Villeneuve's lab, which is focused on studying early functional markers of Alzheimer's disease, studying both proteinopathy and its interaction with brain dynamics. So here's an outline of the talk that I'm going to be giving today. I'm going to start by providing a background mainly about Alzheimer's disease. And I assume many people in here might not be familiar with MEG recording, so I'm going to be giving you a primer about the basic of MEG, does it measure, how we process the signal, and how do we analyze it, just to make sure everyone's on the same page. Then I'm going to do a short commercial about the PREVENT-AD cohort and give you some of the generalities before moving into my 3 research project that integrated my PhD thesis. And finally, I'm going to give you some conclusions, and we can proceed to have a discussion if you have any questions at the end. All right, so moving into the background. As you probably know, Alzheimer's disease is the most common form of neurodegenerative dementia, and it affects over 55 million people worldwide, and some estimates about its prevalence estimate that it's going to almost triplicate by 2050. So it is a very important problem for society, both in terms of the burden that it has for patients, caregivers, but also in terms of the economic burden that it implies for society. Alzheimer's disease features progressive cognitive impairments that appear across multiple cognitive domains. Typically, these start first manifesting in memory and attention, but it later extends to language problems, visuospatial deficits, and disorientation, among other symptoms that the patients can present in the late dementia stages. Importantly, Alzheimer's disease is now conceived as a pathological continuum that includes a wide preclinical phase of the disease during which pathological processes take place in the brain without any manifest cognitive symptoms. So what this figure here is exemplifying is the Alzheimer's disease continuum with time on the x-axis, and you have cognitive function on the y-axis, and you can see how there's an expected decline that is normal with development. As we age, of course, many cognitive functions start declining. But what we see in the Alzheimer's disease continuum is that at some point, people with Alzheimer's start deviating from what is expected to be the normal decline with aging, and they start presenting cognitive impairment. But as I mentioned, there is this extended preclinical phase that can last up to 2 decades before the symptoms appear, where these pathological processes are developing. So it is ideal to study the mechanisms of the disease here, and it's potentially an ideal therapeutic window to intervene in these pathological processes before they cause neurodegeneration and cognitive decline. As you probably know, Alzheimer's disease is characterized by the pathological accumulation of amyloid beta plaques, shown here, and intracellular neurofibrillary tangles made of tau proteins. So you can also see it here in this cartoon, where we have these blobs of amyloid beta oligomers that aggregate into plaques that become toxic to the neurons and compromise the entire homeostasis of the cellular environment. And we have the deposition of tau inside the neurons that eventually leads to neuronal death, which you can appreciate here in what is called a ghost angle. Alzheimer's disease tends to follow a relatively stereotypical pattern of distribution. For amyloid, for example, it starts accumulating in temporal lobe regions and the basal forebrain, and then it starts extending more diffusely across the cortex. And in the late dementia stages, based on neuropathological studies, we see that it's pretty much all over the brain. In contrast, tau deposition starts early in the transentorhinal cortices before extending to other limbic regions and cingulate cortices. And eventually, in the late disease stages, it starts spreading all across the neocortex. And importantly, we now have some imaging tools that allows us to study this deposition of proteinopathy in vivo. So by means of PET imaging, we can also evaluate the deposition of amyloid and tau across the Alzheimer's disease continuum. And most studies have demonstrated that we see a very good correspondence between what has been reported in postmortem studies and what we can measure with PET imaging. Amyloid and tau are part of a very complex cascade of pathological events that eventually lead to neurodegeneration as cognitive impairment. So these processes include, for example, astrocyte activation, loss of synapses, neuroinflammation, and axonal damage. So it is a really complex set of processes that are taking part. And eventually, these are going to lead to neuronal death, cortical atrophy, which is more closely correlated with the cognitive impairment that we see later on. And as I mentioned, these pathological processes are quite slow, so they take place over many, many years before they reach the point where we start seeing deficits in the participants. There has been a lot of discussion in the past years about the biological definition of Alzheimer, because traditionally it was conceptualized more from the clinical perspective. But given that we have an extended preclinical phase with no symptoms, now people are shifting towards defining Alzheimer's disease also based on the biology of the disease. So this is from a paper published last year from the Alzheimer's Association work group, where they did this consensus and proposed a new set of guidelines for definition of Alzheimer's disease. And as highlighted here by these arrows, what you see is that amyloid and tau markers of proteinopathy, both from PET and also from some plasma or CSF markers that we can quantify, are being considered as the core biomarkers for diagnosing the disease. But they have also been proposed to stage the disease severity along with some other non-specific biomarkers of Alzheimer's disease that include, for example, atrophy and hypometabolism quantified by FDG-PET, and some other markers that we can get from MRI, for example, like infarction and white matter lesions that are used to aid, even if they're not specific to Alzheimer, they can aid for the staging of the disease. So this figure, very common on the field, summarizes the time course of the different pathological changes that occur on the Alzheimer's disease continuum. As I mentioned, amyloid deposition starts very early on the disease continuum, accumulating many, many years before we can start detecting tau and other pathological events. And then later in the disease, we're going to see that tau starts accumulating and is followed by neurodegeneration and cognitive impairment. However, it is worth noting that this biological definition of Alzheimer's disease currently does not include any functional alterations of brain activity. And this is something that I'm very interested into and is what I've been working on during my PhD. So it remains the question on how brain activity changes along the Alzheimer's disease continuum. How do these changes relate to Alzheimer's disease proteinopathy? And what are the implications of neurophysiological changes for clinical progression? So just to make sure everyone's on the same page, I'm going to give you some information about magnetoencephalography, which is the technique that I've been using in all of my studies. And MEG is basically a very sensitive, non-invasive imaging technique that allows to detect very small magnetic fields that are produced by neuronal activity. And therefore, it offers a unique opportunity to study brain dynamics, non-invasively, but also with a very good temporal and spatial resolution. So here on the left, you have a participant sitting inside the MEG scanner. And on the right side, you have what would be the viewing of the sensor. In this case, is event-related activity. And you're seeing it from the top of the head. So you see that different traces have higher or lower amplitude, depending on the sensors and the proximity to them. And we can use this spatial information to reconstruct kind of like the magnetic fields and try to reconstruct the sources that are giving rise to this activity that we see on the brain surface. So MEG signals primarily arise from synchronous postsynaptic currents that are generated mainly by pyramidal neurons in the cortex. And this has to do with the anatomy of the brain. So in order to be able to detect these very small magnetic fields that are produced by neuronal activity, we need to grab the information produced by many thousands of neurons that are acting in synchrony. And also the anatomical disposition of these neurons is going to be influencing how sensitive we are to capture their activity. So it has been demonstrated by many research studies over the past few decades that the main contributors of the signal that we see both in EEG and MEG is the pyramidal neurons that are oriented in cortical columns. Because as you can see here, this anatomy allows for the electrical potentials to travel from like up and down, and then we would have a circumrounding magnetic field that is associated with these changes in electrical currents. And when we have many neurons that are synchronized, we can see that there is a summation of this electric potential, or in this case, the magnetic fields. And then therefore we can capture this activity at the level of the surface. So just giving you a comparison between MEG and EEG, you probably heard that MEG has better spatial resolution. And the reason for that is that unlike EEG, MEG is exempt from the problem of volume conduction. So in this figure on the left, you see a cartoon that exemplifies different types of electrophysiological recordings. So you have the cortical surface. And as you know, above this, we have the subarachnoid space filled with CSF. We have the skull and we have the scalp. So when we use EEG or MEG, we're recording these signals from outside the scalp. And in the case of EEG, we're going to be using sensors attached to the scalp. The problem with EEG is that these electrical currents are kind of like diffusely spreading across these different tissues, because these different tissues have different properties in terms of their conductivity. And this is basically make the problem of volume conduction, which means that the electrical currents are going to spread all over the place, and that's going to make it much harder for us to actually identify the sources of this activity. With MEG, we have these sensors that are placed outside the head, of course, and are not attached to the scalp exactly. But they have the advantage that given that they're recording magnetic fields and not electrical currents, they are exempt of this problem of volume conduction. So all of these different issues would be kind of like invisible for these magnetic fields, and therefore we have a better signal to noise ratio, and we have better precision for identifying the sources that produce this activity. So this is just a figure from a review from my supervisor in 2017, where they show the comparison between the resolution that we can get from EEG and MEG. So as I was mentioning, you can see that it is complementary signals that we see in EEG and MEG, depending on the structure of the cortex. For EEG, for example, we're going to be more sensitive to the sources that are aligned radially, respective to the surface of the cortical structure. And then for the MEG, we're going to be more sensitive to the neurons that are oriented tangentially. So the neurons that are inside the brain sulci are going to contribute more for MEG signals. So it's not exactly the same signals that we are capturing, but it's kind of like complementary measures. But the advantage with MEG is that, as I mentioned, given that we are exempt of this problem, we can have better localization. I don't know if you can see it from here, but this is showing where the actual source is based on reconstructed artificial data. And with EEG, you can see that there's some degree of error and also some bias in terms of the amplitude. So in conclusion, MEG has better spatial sensitivity compared to EEG because of these reasons. So for acquiring MEG data, we need a lot of technical equipment. The sensors that capture these magnetic field fluctuations have to be extremely sensitive, because the order of magnitude of MEG signal is around the order of femtoteslas, which is 10 minus 14 teslas, which is very, very, very small. So basically, we need special type of sensors that are called squids. These are the ones that are used more commonly with the available technology that we have today in most settings. And it's basically a series of sensors that are cooled down with helium to be at a superconductive temperature in order to be sensitive enough to measure these slight variations of magnetic fields. And additionally, we have some reference sensors inside the scanner that are going to be helping us to record the noise that is coming from outside the head, everything that is outside the brain, and it's going to help us with the processing of the signals, because even if we have like an insulated room and a magnetically shielded room where we put the participant and the scanner, we're still going to be capturing environmental noise coming from different sources, like even the computers, the equipment from the hospital. So it does require a lot of specific hardware, and also like the expensive room of like being magnetically shielded so it can be recorded properly. We also need some sort of way to digitalization the head shape of the participants. So we in the MNI have a system that allows us to use different kind of like virtual points to measure the head of the shape of the head. And this information we're going to be using to make an accurate registration to the structural MRI of the participants in order to improve our spatial resolution and our precision to identify sources. And finally, we also use other types of physiological recordings because some of these physiological processes can also induce artifacts on the MEG signal. So for example, muscular activity or the heartbeat itself, it's several orders of magnitude bigger than the MEG signals. So it can compromise the MEG signals, and we're going to be constantly dealing with this kind of artifacts. So I would say this is the main challenge about MEG, that we're trying to measure something that is very, very small, and we need to get rid of all of the noise that is produced both by the body, but also by the environment that is surrounding the participant. In terms of the processing of MEG data, the first step is to register the MEG signals and the position of the sensors relative to the anatomy. So you can either use a default template that it can be an average across many subjects, but typically we want to use the individual specific anatomy to have a better mapping of the actual, like shape of their brain, the size of the head, the form of the sulci and gyra, which is all going to help us to have better spatial resolution for identifying the sources of MEG. We're going to have to filter the data, which is going to help us to deal with the artifacts that we have coming from physiological and non-physiological surface. So we typically use like a high-pass filter to remove low frequencies that are associated with these artifacts coming from environmental noise. And we're also going to be filtering the power line, which also creates a very large amplitude artifact on the MEG signal. This is very constant. So here in North America, it's like 60 hertz. In Europe, it's 50 hertz. And it's relatively easy to remove because we know the exact frequency and we can just like take it off the raw signal that we have. As I mentioned, we need to identify some events related to cardiac activity and blinks, and therefore we use EEG sensors or different types of electrodes to record the signals, and that's going to help us during the pre-processing in order to modulate and kind of like estimate the distribution of how this artifact looks and then kind of like subtract it from our signals. By constructing a model that is adapted to the individual anatomy and produces kind of like a surface, we can use this for estimating the forward problem, which would be basically trying to resolve like what are the sources that are generating the activity that we see like outside the brain. So we need a mathematical model that kind of like infers based on what we're recording at the surface, where is it most likely that it is coming from, also accounting for the shape of the head and different properties of the tissues that we can incorporate. And lastly, the last step is kind of projecting the data into what we call source space, which is basically bringing the MEG signals from the sensor space and projected on the cortical surface of each participant. So we can end up having like a brain map very similar to the ones that we see in other types of functional imaging like fMRI, where we're going to have a single time series for different vertices of the cortex, or we can also average the vertices across different regions based on a parcellation, and therefore we can extract this time series, but already on the cortical surface rather than in the sensor space. In terms of the analysis, MEG are typically analyzed in the frequency domain. So we can quantify what is called the spectral power or the energy that different frequency bands has in order to study brain dynamics. And different frequencies have been associated with different cognitive processes and with different physiological states. So here on the left, you have an example and a cartoon of a typical fast Fourier transform. So we have a complex signal that is integrated by different frequency bands, and with the Fourier transform, what we can see is the exact contribution of different frequencies to this complex signal. And when we move it from the time domain to the frequency domain, we can quantify this as a power spectrum, and therefore we can have an objective measure of how much each frequency band of the spectrum is contributing to the signal. And this we can use, as I mentioned, to study normal cognitive processes. For example, during sleep, the delta waves, which are the slower in terms of frequency, are more predominant during this deep sleep. The alpha waves are the most prominent in the awake EEG or MEG recording when we see participants in a relaxed state or with their eyes closed. And we have faster rhythms such as beta and gamma when participants are awake but alert, or when they are performing a cognitive task. So even if we have different proportions of these rhythms, this is going to be changing dynamically all the time. And it is important to understand that all of these frequencies are kind of like present and embedded within the complex MEG signal. It's not that one appears and the other disappears. So we're going to have a complex mixture of all of them, and it's going to be very dynamic so that it changes even in the scale of milliseconds. We can also use MEG to study the temporal relationship between the activity recorded in different brain areas, and we can derive different types of measures of functional connectivity based from the MEG signals. So most of these connectivity analyses are based on either the amplitude or the phase of the sequences. So here in this figure, you can see, for example, in the left, what would be a type of functional connectivity based on the amplitude. So you see that even if the signals have different frequency, the amplitude of these signals is well aligned between one another. So we can estimate their connectivity based on these amplitude fluctuations, or we can also study how much they are synchronized based on the phase of the different oscillations. So as we do with fMRI and other techniques, in the end, we can have a reconstruction of a functional connectivity matrix that is going to be summarizing the statistical dependencies of the time series, either in the sensor space or, if we want, in the source space once we already projected that into the cortical surface, which is what most people do in order to take advantage of the resolution of MEG. So as I mentioned, I've been working with the PREVENT-AD cohort since I started my PhD. Probably you've heard about it, but just to give you an overview, the PREVENT-AD is a longitudinal observational study that started in 2011, and it enrolled 387 individuals with a family history of Alzheimer's disease. This was defined as having either one of their parents or two or more of their siblings diagnosed with Alzheimer's. And they were also required to be older than 59 or above 55 years old, if that age was within 15 years of the age of onset from their closest relative that had the diagnosis of the disease. In addition, none of the participants had any other diagnosis of neurological or neuropsychiatric disorders at the time they were enrolled into the study. And very importantly, all of the participants were cognitively unimpaired when they were enrolled into the study, and at the time they had their first imaging session for the PET and the MEG, which is what I'm going to be talking about today. And this was determined at an eligibility visit where they underwent some cognitive screening in order to ensure that, in fact, they were all cognitively unimpaired. This is a figure from a paper that we published this year, kind of like summarizing the data that is available in the PREVENT-AD cohort, which is now openly accessible for all of you. So I'm going to take this couple of slides also to promote the use of the PREVENT-AD data. So here is an overview of the different modalities that we have available. As I mentioned, we have multiple imaging modalities comprising structural and functional MRI. We have amyloid and tau PET scans from these participants. We have the MEG, but we also have a lot of behavioral data about cognition, sleep, neurosensory data, and we have other types of biomarkers captured in the CSF or in the blood, which have been demonstrated to be very robust for detecting Alzheimer's disease pathology. And importantly, in this open release of the PREVENT-AD, we also include different data derivatives, which is kind of like analytic measures that people can already use for different types of analysis and for responding different scientific questions without having to undergo through all of the pain of like pre-processing the data, doing the analysis. And here you can see also the different types of behavioral questionnaires and biofluid measures that we have. So I will refer you to the paper in the end in case you are interested in looking more in depth the data that is there available. So this figure is just kind of like summarizing the timeline for the data acquisition. This is for the cognition. And as you see, there are participants who started even in 2011, 2012. And some of them have now up to 10 or even 12 years of longitudinal cognitive follow-ups, which is very valuable, especially when you're tracking participants when they were still cognitively unimpaired to the point where some of them are actually progressing. Here is for the imaging sessions. So the data that I've been using mostly is the PET and the MEG, which was collected in this window between 2017 and 2019. And we also have the fluid biomarker data that started being collected in 2012 and is still ongoing data collection. Importantly, one very interesting feature of the PREVENT-AD study is that given that we have been following these participants for many, many years, we now have some participants who have progressed to MCI or to develop cognitive impairment and dementia itself. So this is a figure from the paper that is basically showing the rate of progression of people based on their proteinopathy profile. So for people who have no amyloid and no tau in their brain, we see a very low rate of progression from cognitively unimpaired to MCI. Whereas for people who have high levels of both amyloid and tau in this group, we see that around half of them were progressing after 3.5 years of follow-up. And after 6 years of follow-up, we have that all of these participants eventually progressed to MCI and to dementia, which is what would be expecting, of course, from previous studies. So here's the paper. I encourage you to go and have a look, and maybe you will find some interesting data that you can incorporate into your own projects. And you can reach out to either me, Sylvia, or any of the Villeneuve lab members if you want to have some more information or some help in accessing the data. All right, so moving now into my research itself. Here I'm summarizing the data that I used during my PhD. So I started in 2020. And the data that I've been using mostly is the baseline resting state MEG and amyloid and tau PET scans that were collected in this window, 2017 to 19. But I also use some of the imaging data on some plasma measurements that we have. So I'm going to be coming back to this slide to show you the data that I use for each of my PhD projects. All right, so moving into my research questions. The first project aimed to assess whether the pathological deposition of amyloid and tau is associated with changes in neurophysiological activity measured with MEG during the preclinical stage. So that brings us to the first chapter of my thesis. So amyloid deposition has been demonstrated to be linked to neuronal hyperactivity. This has been demonstrated by animal models where they use genetically modified mice that overexpress either amyloid, tau, or both of them. And you can do different types of electrophysiological recording, either at the level of single neurons or local field potentials, a group of a few thousands of neurons. And what people have discovered is that neurons that are close in proximity to amyloid beta plaques show this hyperactive pattern, which is defined as an increase in their firing rate as compared to a baseline state in a control mice with no pathology. And later on in the disease, interestingly, when we have the presence of high levels of both amyloid and tau deposition inside the neurons, we see that there is a shift towards suppressed neuronal activity. So it goes from like normal firing rate to hyperactivity to then hypoactivity. And then it's then when we see this suppression of neuronal activity that we also start seeing behavioral deficits in the animals. So there has been a lot of research, of course, using EEG or MEG in Alzheimer's disease patients, and they have consistently demonstrated that in the late stages of the disease, there is a generalized slowing effect. That means that the proportion of slow frequency waves in the resting state MEG or EEG becomes much higher as compared to the faster frequency oscillations. So this is what they call slowing. And this is well correlated with the late disease stages of late MCI and Alzheimer's disease dementia. And it has been also demonstrated that the more slowing these spontaneous oscillations become, the more severe are the cognitive deficits of the participants. However, given the challenges of studying the preclinical stages of the disease and recruiting participants that are on the path of Alzheimer's disease but do not have symptoms yet, we still don't fully understand how those neurophysiological activity change during the preclinical stage. So my first project took as a starting point the evidence coming from animal literature. And what I wanted to test is whether we can see analogous shifts in frequency-specific activity that reflect some sort of hyperactive or acceleration effect on MEG signals during the preclinical stage of the disease in association with amyloid deposition, and whether we can observe also a shift towards hypoactivity that relates to both amyloid and tau deposition as suggested by the animal literature. So the aims of this study was to test the association between neocortical amyloid and temporal tau deposition on frequency-specific spectral power of MEG resting state recordings. And we also wanted to see whether these changes in neurophysiology that are associated with proteinopathy also predict longitudinal cognitive decline of the participants. So for this project, I used the MEG and PET data that was collected during the baseline. I also used the cognitive assessments, both retrospectively and subsequently acquired after the imaging sessions, and the T1 weighted imagings to assist for the PET and the MEG processing. And the final sample size for this study was 104 participants. So this figure summarizes the multimodal data processing and analysis pipeline. So in terms of the MEG, as I mentioned, we go through the pre-processing of the signals first in order to filter the data, get rid of the artifacts, do the alineation with the MRI, then we can project these signals into the source space or into the cortical surface. And we can align this with the MRI and use different parcellations based on the type of analysis we want to do. And in parallel, for the PET imaging analysis, we pre-process the data, we align the images, we correct for motion, and we can quantify the tracer uptake based on taking a reference region that has no amyloid and tau deposition in theory, such as the cerebellum. And then we can have a quantification of the tracer binding that is going to be reflecting the amount of amyloid and tau that we have in different brain regions. So for my project, I focused specifically on 4 canonical frequency bands from the resting state MEG activity, which are delta, theta, alpha, and beta, ordered based on their frequency, as you can see here. And for the amyloid and tau, I focused on the whole brain regional amyloid deposition and tau deposition in the entorhinal cortex and the temporal lobe. As we know, this is one of the places where tau starts accumulating very early on in the disease continuum. And given the early stage of our participant sample, we wanted to focus on the temporal lobe for assessing tau pathology.
Can you tell me a little bit more about how you go from your surface level MEG data and register that to the T1 weighted, or what you're registering or what you're transforming to get it in the same space?
Yeah, so during the MEG data acquisition, we bring the participant to the scanner, and before putting the participant inside the scanner, we have this digitization system. I don't know how to call it with another label, but it's basically like a pen that we use, and we mark different points in the head so we can have a reconstruction of the head shape of the participant. And we also mark some anatomical landmarks such as the nasion, the preauricular points, and also the different coils that we use for recording movement inside the scanner. So now that we have this digital shape of the head, we grab the MRI, we import it into the software, and then we reconstruct not only the cortical surface, but also kind of like the mesh of the head.
Yeah.
And then we use the spatial information from what we collected during data acquisition to align it with the head mesh.
It's like you have tags from the skull or from the outside of the head.
Yeah. So you import this file, this position file that has all of these coordinates for the different points that we mark. We typically do kind of marking points all spread across the skull. And this information is what we use for aligning it with the MRI.
Thanks.
Yeah. Sure. All right. So for the actual statistical analysis, I wanted to test the association of proteinopathy with neurophysiology. And the way we did it was using nested linear mixed effect models. So we have the deposition of amyloid across the 68 regions of the Desica and Kiliani atlas. And we also have spectral power measurements for each of these regions that we extracted from the reconstructed MEG signals. And we're going to be trying to model the association between amyloid deposition and MEG spectral power within each person brain. So here, each of the lines represents one participant, and each of the dots would be a specific region of interest. So we are going to be modeling the within subject association between regional amyloid deposition and regional changes in spectral power of MEG. And then in order to test whether there's an interaction effect with tau, and whether it produces change in this association between the deposition of amyloid and spectral power, we included tau deposition in the temporal lobe as an interaction term in order to see whether people who are accumulating more tau are showing this shift that I was mentioning. We hypothesized based on the animal literature. And for the subsequent analysis, we try to assess whether these changes in neurophysiology associated with amyloid, but also with tau, are kind of related to longitudinal cognition. So we take this within subject association of the amyloid deposition and the neurophysiological changes, and we incorporate tau again as an interactive term, and we try to predict the longitudinal cognitive slopes based on this state of the pathology of the participants. Yes?
Question. But you're looking at the baseline. So you have one shot, but you have a time series, and you have the frequency domain. And you think change within a session, change compared to the reference point?
It wouldn't be the change. So we only have this one measurement of MEG. We have 5 to 10 minutes of signal. We eventually remove some epochs and everything. But in the end, what we do is we quantify the spectral power for different time segments. These are typically 4-second epochs that we use in order to get rid of the epochs that have artifacts. We quantify the spectral power for each of these windows. But then in this case, we're just averaging out across all of the epochs. So we're not looking at the change dynamically. We can do that with MEG. But in this case in particular, we're summarizing the information as a single number that is going to be representing the overall spectral power across the 5 to 10 minutes of the recording.
By changing physiology, you mean cross-population association?
Yeah. It would be more the change over time based on what we're predicting. Yeah.
But you have only one time point for physiology.
Yeah, no, when I was referring to the changing neurophysiology across the Alzheimer continuum is more like what has been defined like in the literature and what we would expect to see over the course of many years. But yeah, in this type of analysis is just cross-sectional data that we have. So it's a single MEG measurement against a single measurement from amyloid and tau PET. Yeah. All right, so this first figure summarizes the main result that we got from these projects. So the hypothesis was that amyloid beta was going to be related with expressions of acceleration of neurophysiological activity, and this is in fact what we found. So here at the top you have a map that shows the distribution of amyloid beta across the brain. Here you have the distribution of spectral power for the slow delta frequency band and for the faster alpha frequency band. And what we observed interestingly is that regional changes in regional activity in the delta band was negatively associated with the regional binding of amyloid tracer, whereas there was a positive association with alpha activity. So this means that amyloid beta is related to an enhancement of faster alpha activity, which has been associated and modeled by some computational modeling studies as a potential reflection of hyperactivity at the macroscopic level. And interestingly, when we include tau as an interaction term into this model, we start seeing that individuals that accumulate higher amounts of tau starts showing a shift in this association. So eventually it will reach a point where you have so much tau that the relationship is going to become negative, meaning that with higher amounts of pathology, you're going to be having expressions of neurophysiological slowing, meaning like increases in delta and decreases in alpha.
Would you expect this relationship to be linear?
Yeah, it's complicated because like if we were to like hypothesis across the entire AD continuum, we expect to see that it is like in fact non-linear. Like I tried to do some non-linear modeling in this specific approach, but it's complicated given that we have like a single time point. So when assessing the association at the baseline level, we do see that the linear association fitted better. But the fact that we're seeing a shift with the tau interaction, which is an indicator of disease progression, like we know that people who have more tau are probably later on in the disease continuum. So we are starting to see this shift. So it's kind of like telling us some information about like the evolution of the continuum. But of course, we would need longitudinal data in order to actually test this empirically and see whether it is actually changing, which is one of the things that we're working on right now that we have already some longitudinal MEG available.
And at baseline in Prevent AD, do you think that everyone, or can you speak to where people fall on the like AD continuum? Because based on the inclusion criteria, if I remember correctly, it's supposed to be kind of an early phase, right?
Yeah.
But you do see people with kind of this variance in how much amyloid and tau is already accumulating, right?
Yeah, exactly. So at the time they were enrolling to the study around 2011, 12 for most of the participants, they had like a neuropsychological assessment. So they verified, okay, all of them have family history, but they're still cognitively unimpaired. And then they have like longitudinal cognitive assessments every year. So we know for all of the participants that I included in my study that at the time they have the MEG and the PET measurements for the first time, they were all still cognitively unimpaired. For the longitudinal PET and longitudinal MET data that we have available, we know that some participants have now progressed to MCI and some of them even to dementia. So it's going to be very interesting to look at the longitudinal MEG data to see if that in fact we're seeing this change over time while we're measuring the same participants across time. So in terms of the association with cognitive performance, as you can see here in this plot, we found that as expected, people who have high levels of both amyloid and tau are showing steeper negative slopes in terms of cognition. Here we're measuring specifically the composite score from attention and memory tests that are included in the Arban's neuropsychological battery. And as I mentioned, the ones that have both amyloid and tau are the ones that are showing decline, which is in line with previous studies that have shown that there are people who have high levels of amyloid, but they do not necessarily show cognitive impairment, so that you need both amyloid and tau to start seeing this decline. So the last analysis that we did for the cognition was to try to assess whether this shift in neurophysiology is associated with cognitive decline. So here in the x-axis, what we're seeing is the amyloid alpha association. So a higher correlation would be indicating more alpha with higher amounts of amyloid, and a lower correlation would be the opposite pattern. And we're color coding the participants based on their amyloid and tau status. So the red group is the one that has the higher level of tau. This is just for the purpose of showing the interaction, but in the analysis, all of the data was used as continuous values. But what you can see here for the people who have high levels of amyloid and tau is that the ones that are showing a more negative alpha amyloid association, being the ones that are showing a more pronounced shift in the associations that I show, are the ones that are showing the steeper cognitive decline. So this all goes in line with the idea that it is the shift towards slowing that is predicting that participants are going to decline longitudinally in terms of cognition, and not only the acceleration effect that is associated with amyloid, because in the participants that have amyloid but not tau, we don't see this association. And this figure is another way to visualize this interaction effect. So this one I really like because you can very clearly appreciate the interactive effect and the hyperactivity promoted by amyloid versus the hypoactivity promoted by tau. So if you see the x-axis of the figure, you have the global amyloid deposition expressed as centiloid units. And in the y-axis, you have the tau SUVR quantified in the temporal lobe in a meta region of interest that not many people use in the field. And the color map represents the change, the relative alpha power, So what you see here is that when you have low levels of tau and we have increasing amounts of amyloid, we see that it is accompanied by an increase in alpha power, reflecting potentially like acceleration of brain activity or hyperactivity. And then when we see increases in amyloid, but also increases in tau, we see that there's a shift towards lower alpha power. So this is a very nice way to visualize the interaction effect. And lastly, what we did in this paper was to compare the prediction of our models, which is basically this heat map, how much this is aligned with empirical measurements of neurophysiological activity in MCI and AD patients. So this cross that you see here in purple are the amyloid and tau values that were quantified in a large sample of MCI and AD participants. So the people from Prevent AD have very low amounts of pathology, they fall in here, and the people with the later disease stages have, of course, higher levels of amyloid and tau. But what this plot is representing is that based on the predictions that we did from the neurophysiological shift in the preclinical stage, we can accurately predict how most neurophysiology is going to shift. And we see that there is a very good alignment with the estimate of the shift in neurophysiology represented by this black line and the confidence intervals. And it predicts very well how much the alpha is going to be shifting based on the amount of pathology that we know people in the later disease stages have. So basically, we see that the predictions of our model conducted in the preclinical stage kind of like aligns well with the amount or the magnitude of the slowing effect that we see actually in patients in the later disease stages. So in summary, amyloid deposition enhances alpha activity, which might be considered a proxy of neuronal hyperactivity, while medial temporal tau deposition promotes a shift towards slowing of brain activity. These proteinopathy-related shifts are associated with longitudinal cognitive decline in attention and memory, and are well aligned, as I just showed, with empirical reports coming from MCI and AD patients. So moving into the second research question that I addressed during my PhD, here we wanted to test whether we can use functional and structural connectivity information in order to predict patterns of longitudinal tau accumulation. So just to provide you some background, this project was motivated by the fact that there is a lot of evidence, again, coming from animal models that have demonstrated that tau pathology can spread across synapses and propagate from early tau accumulating regions such as the entorhinal cortex, or as many people call them, tau epicenters, to other cortical regions that are strongly connected both by anatomical pathways, but also it has been shown with functional connectivity. So animal research using, again, transgenic mouse models have also demonstrated that the presence of amyloid beta seems to be facilitating this effect, and it dramatically increased the speed and also the extent of the tau propagation. So when you have like mouse models that overexpress tau but no amyloid, you see some spreading of tau, but it's mainly delimited to the temporal lobe and the hippocampus and surrounding regions. But when you also have mice that is expressing also high levels of amyloid, you see that the spreading of tau kind of like goes crazy and start like going all over the place much faster than compared when we don't have amyloid. And there are some studies in human participants that have also supported this kind of synergistic facilitation of amyloid beta deposition, because we know that there are some people with neuropsychiatric conditions that express high levels of tau without amyloid. This is called partially age-related tauopathy. That's a good example where people have high levels of tau but no amyloid. And in those participants, the tau spreading tends to be delimited to the temporal lobe and the cingulate cortex, as exemplified here in red. Whereas in Alzheimer's disease, we have that tau starts spreading across all of the rest of the neocortex. So this more pink tone coloration of the different brain regions that you see here, it would be kind of like the tau spreading that is facilitated by amyloid beta. So there's some evidence coming from both animal models and from studies in human patients that kind of suggest that tau can spread across brain networks, but also that amyloid plays an important role in facilitating this process. So the aim of this study was to use structural and functional connectivity methods derived from multimodal neuroimaging in order to assess whether connectivity can predict the longitudinal accumulation of tau, and whether this is modulated by the amount of amyloid that this participant express. And we also wanted to test whether this connectivity facilitated accumulation of tau would be related to clinical progression to MCI, because now that we're using the longitudinal tau, we know, as I mentioned, that some participants are actually developing cognitive impairment. So the data that I use for this project is, again, the baseline amyloid and tau PET data and the MEG that were collected in this time window. But for quantifying longitudinal tau accumulation, now we're looking at the rate of change between the baseline tau PET visit and the follow-up tau PET visit, which was acquired around 4 to 5 years after the first one for most of the participants. And in order to quantify functional and structural connectivity, I would be using the resting state MEG. Importantly, at the time, the participants were still cognitively unimpaired. And I also derive functional connectivity measurements from fMRI and structural connectivity measurements from diffusion-weighted imaging that we also have from these participants. And in order to define the cognitive status of the participants, there is a team of neuropsychologists, experts from the PREVENT-AD group that basically kind of like do a periodic review of the clinical history of the participants, their neuropsychological assessments, and based on this criteria, they define whether people have progressed to MCI or whether they're still cognitively unimpaired. So this is information I'm going to be using to see whether people who progress to MCI have a stronger accumulation of tau over time. So as I mentioned, we have 3 different imaging techniques in order to assess structural and functional connectivity. So I'm not going to go very deep into the details for the processing of diffusion and fMRI. But basically, as you know, we can use diffusion-weighted images to quantify a whole brain tractogram that is going to be showing us the structural pathways that connect different brain regions. We can then filter these tracks in order to construct a structural connectivity matrix based on a specific parcellation of our choice. In this case, I use the Schaefer 200 atlas. We can do the same for functional MRI by extracting the time series from different parcels and then computing the Pearson correlation coefficient in order to derive, again, a functional connectivity matrix that shows the associations across all pairs of these Schaefer atlas regions. And as I mentioned in the introduction, we can do something very similar with the MEG signals, which is kind of like what we do with the fMRI. We extract the time series, we choose our measure for assessing functional connectivity, which in this case, I use a technique called amplitude envelope correlation, which is a measure that is based on the amplitude of the signals to assess the functional connectivity. And in the end product that we have is a functional connectivity matrix with the same dimensions that we have for the other modalities. So this is the number of participants who have this data available, and the final sample size that I used in this project was 81 participants who were the ones who had all 3 imaging modalities available for deriving connectivity estimates. In terms of the proteinopathy burden, we quantified global amyloid deposition by taking a weighted average across all of these regions, because we know amyloid accumulates more diffusely across the brain as compared with tau. And for defining tau epicenters, we did that at the individual level. I'm going to explain more in detail this approach in the next slide. But importantly, keep in mind that we define the epicenters at the level of each individual. And for quantifying longitudinal tau accumulation, as I mentioned, we measure the annual rate of change between the follow-up and baseline scans in order to have an estimate of how much tau people are accumulating every year. So this is the main analysis approach that I use for this project. So in order to define the epicenters for each participant, which was the first step, we sorted the 200 Schaefer atlas regions based on the baseline tau SUVR, and we selected the top 10% or 20 regions that had higher baseline tau SUVR. So for example, it could be that we selected these 20 regions. Then we go to each of our connectivity matrices. This is done separately for each modality at this stage. And we extract the connectivity vectors corresponding to each of these 20 regions, which is exemplified here. And then we just compute an average across the columns in order to have a single vector that is going to be representing the strength that each cortical region, the strength of the connectivity of each cortical region with the tau epicenters on average. And then we use that information in order to try to predict longitudinal tau accumulation. So again, I use linear mixed effect models in order to see whether the structural and functional connectivity was associated with tau accumulation. And we incorporate the global amyloid burden as an interaction term to see whether the amount of amyloid is going to be modulating this association between connectivity and tau accumulation. So this figure exemplifies the main result that we got from this project. So as expected, based on our hypothesis, we do see that there is a positive association where stronger structural but also functional connectivity predicts increasing amounts of longitudinal tau accumulation. But importantly, we see a significant interaction effect where it's only the people who have high levels of amyloid beta, the ones that are showing this strong positive association. So again, in the models, we also used continuous values for amyloid deposition and longitudinal tau accumulation. And just to show the interaction effect, I'm splitting the participants based on the top quartile of participants that had higher levels of amyloid versus the rest of the participants. And we see very clearly here in this plot that it is the participants who have high amyloid, the ones that are showing this association between strength of connectivity and longitudinal tau accumulation. And importantly, we see consistent results across all three imaging modalities. Mainly, we see stronger associations for the structural connectivity derived from diffusion-weighted imaging and the functional connectivity in the alpha band specifically, which is also aligned with what other studies have demonstrated in the preclinical and the symptomatic stages of the disease. And I forgot to mention here at the top, we have a map that shows the distribution of tau epicenters across participants. So you have the scale from 0 to 81 participants, which is the total amount that we have. And you see the color scale on each region represents how many participants had that region selected as a tau epicenter. So we see many regions in the temporal lobe and close to the posterior cingulate cortex that were defined as epicenter across many participants, where some other regions were not selected as epicenters in any case. And this distribution, it's kind of like very similar to what other people have reported at these early stages of the disease. Then we wanted to see whether integrating multimodal information can allow us to predict better longitudinal tau accumulation. So we basically did a weighted sum across the different connectivity modalities in order to generate a multimodal connectivity matrix. And basically what we found is that we see even a stronger association when we integrate structural and functional information, where we also see this interaction effect that individuals with high amyloid show higher tau accumulation. And lastly, what we demonstrated, if you look here at the epicenter connected regions based on multimodal connectivity, is that people who progress to MCI show higher levels of tau accumulation in the regions that are more strongly connected with the epicenter as compared to people who remain cognitively unimpaired. So all of this is kind of like supporting the idea that functional and structural connectivity is related to tau accumulation and that this is modulated by amyloid beta, and that longitudinal tau accumulation in these regions that are connected to the epicenters is associated with clinical progression to MCI. So lastly, I'm going to go very quickly through the last project. So the idea was to test whether neurophysiological activity can complement established biomarkers for predicting MCI progression. So At the time I did this analysis in 2024, we had around 30 participants in the PREVENT-AD, 30% of the participants that progressed to MCI over 7 years of longitudinal follow-up from the first PET scan to the last cognitive follow-up in 2024. And therefore, we can try to assess, because we have biomarker measurements before they progress, we can try to assess what are of these biomarkers we can use in order to predict who's going to develop cognitive impairment in the future. So the idea was to test whether neurophysiological activity can complement established proteinopathy biomarkers from PET and plasma for predicting who's going to progress to MCI. And importantly, we wanted to test whether the effect of these biomarkers on the risk of progression is changing over time. So this is the data that I use. I'm going to be looking at the baseline measurements of amyloid and tau, the resting state MEG spectral power quantifications, and also the plasma measurements that were applying probes to this imaging session. So this is the analysis approach that we took. We used Cox regression survival analysis model, so that we started with a basic clinical model, including only demographic information, namely the age, sex, years of education, and also the genetic APOE status of the participants. And then we subsequently combined these demographic features with the different biomarkers that we have available. So the second model incorporated the MEG estimates, in which we focus in the alpha power based on the results of my previous projects. For MRI, we extracted the hippocampal volume. For plasma, we quantified the amyloid beta 40/40 ratio and the p-tau217, which are known to be robust biomarkers of proteinopathy that we can quantify in plasma. And models 5 and 6 combine this clinical information with amyloid and tau, respectively. And we also had a model that combined all of the different biomarkers. So these are the survival curves that we obtained from each of the models. And as you can see here on the stats, or maybe not, we do see that incorporating biomarker information to the clinical model improves the stratifications of participants based on the risk of MCI progression. And this can be better appreciated when we separate the participants on tertiles based on the predicted risk scores derived from the model. So you see that in the clinical model, we have a moderate separation between the curves of the participants that are assigned a high risk score versus the one that are assigned a low risk score. And as we see that when we incorporate information mainly from proteinopathy biomarkers, we have a better separation of these curves, which indicates that the people who are in fact progressing to MCI are being the ones that are assigned the higher risk scores. So the better separation indicates a better discriminative accuracy of our models. And this is just to show the model that combines all the different biomarkers, where we see the strongest effect, and we see the better separation and identification of people at risk of MCI progression. So this plot just shows the contribution of different biomarkers, and I did this basically correlating the raw biomarker measurements against the predicted risk scores based on the Cox model. So as we would expect, being older, being female, being an APOE carrier, having higher levels of alpha power, and higher levels of proteinopathy and PET and plasma were all associated with having higher chances of progressing to MCI, which is consistent with what other people have suggested in the past. And one of the most important contributions of this project is that we were able to show that there is a time-varying effect of neurophysiological activity and also the amyloid beta burden on the risk of progression. So we can start looking at the risk associated with amyloid PET burden, and we see that the hazard risk associated with this biomarker is increasing over time. So in order to quantify this, we added time interaction terms in our Cox regression models. And what this is telling us is basically at different time points, like how much is the risk of you progressing to MCI if you have high levels of amyloid. So if you have an amyloid PET scan and you have high levels of amyloid, you do not necessarily have an imminent risk of progressing to MCI. Even like 2-3 years after, your risk of progression is still relatively low. But if you have high amyloid at baseline, the risk of you progressing to MCI is going to be becoming higher and higher as time goes by. And interestingly, we see the opposite pattern with MEG, which again further supports this idea of a nonlinear trajectory and a neurophysiological shift from hyper to hypoactivity. Because if you have the MEG at time point 0, we see that higher levels of alpha power were associated with a higher hazard risk of progression to MCI. So initially, you have that higher alpha power is going to put you at risk of progressing to MCI. But if we keep following the participants over time, we're going to see that this association eventually is going to shift, so that later on in the disease continuum, lower alpha power is going to become the strongest risk factor. So this goes well with the idea that there's an increase in alpha followed by a decrease in alpha, and that this can be potentially reflecting a functional biomarker of brain function that is changing dynamically across disease stages. And lastly, here I'm just showing that when we incorporate these alpha power fluctuations, these alpha power time varying effects along with the plasma and PET measurements, we see an improvement in our models, which suggests that neurophysiological activity changes can potentially be a complementary biomarker to establish proteinopathy biomarkers. So in summary, we see that PET and plasma are already very good at identifying individuals at risk of NCI, but we do gain some additional information when we incorporate neurophysiological activity, especially taking into account that the risk associated with alpha power is not going to be homogeneous over time and it's going to vary. And when we compare these models with and without including neurophysiological activity, we see better performance, supporting the idea that although it is not a specific biomarker of Alzheimer's disease, such as the quantification of proteinopathy, it might be a complementary biomarker that can boost the accuracy of the models already incorporating clinical and proteinopathy information. So just to conclude, in the first project, we demonstrated that there is a shift in neurophysiological activity, mainly captured by fluctuations in alpha power that are associated with the deposition of amyloid and tau, and that this predict longitudinal cognitive decline and are aligned with what has been described in later disease stages. We demonstrated in chapter 2 that stronger connectivity, both functional and structural, between cortical regions and tau epicenters predict higher tau accumulation in the former cortical regions, and that this is also relating to higher chances of progressing to MCI. And lastly, for the third project, we demonstrated that modeling dynamic trajectories of neurophysiology can provide additional information to inform models aiming to predict the risk of MCI progression. This is just the take home, and I want to finish by thanking all of the people that have helped me throughout my PhD and my projects, specifically my supervisors, Dr. Sylvia Villeneuve and Sylvain Baillet, Dr. Alice Bisman, who contributed a lot to the research work I presented today, my lab mates, some of them are here present in this room, my academic committee examiners, the IPN, all of the people from the staff of the PreventAD research group, the participants, and thank you all for your attention.
So, actually, does alpha power change with age?
Yeah, that's a good question. So we do see that these different brain rhythms are kind of like evolving along the developmental trajectory. Like from like newborns, the spontaneous activity that we see is very different from what we see in adults. So at the beginning of life, in the early stages of development, you see the predominance of more like slow frequencies in the EEG and MEG. Is mainly kind of like theta rhythm frequency between 4 to 8 hertz. And then as people start maturing and reach like adolescent and early adulthood, you see that this baseline theta rhythm kind of like accelerates a little bit and becomes like the typical alpha that we expect to see in normal EEG and MEG spontaneous recordings in adults. This kind of like stays more or less stable until late adulthood. But then during dementia, people have during normal aging in older adults above like 60, 70 years old, they have demonstrated that there is kind of like a progressive slowing effect, but it is not as pronounced as what we see in dementia. Not only in Alzheimer, but in also other neurodegenerative conditions, we see a similar slowing effect. So it is kind of like similar to what we see in terms of like the cognitive trajectory of the participants. Like we do expect to see some degree of cognitive decline with age, which is normal, but in Alzheimer, it kind of like goes down much more faster. And within neurophysiology, we kind of see an analogous effect. Like we do expect to see some sort of slowing of spontaneous activity over time, but in Alzheimer's disease, like it goes down much more faster.
Because the curve you're showing, let's assume that is the curve. People go up and then go down. Mm-hmm. If I take two people at the same level, one of them before, one of them after, do you think based on the rest of the brain, you can say this person is before or after, based on other frequencies and based on the general pattern of the brain? Or you can't even differentiate very easily?
Yeah, I mean, that's one of the things that is very challenging because as you mentioned, like if we follow people along the curve, like it's hard to tell whether you're here or here, you might be going up or you might be already going like towards the slowing effect. So this is why I think, like in clinical practice, if we were to bring this as an actual application for like screening participants, we do have to take this with a grain of salt and assume that these changes in neurophysiology might not be a very specific marker of disease progression when used as a standalone tool. But if we combine it, for example, with the plasma, where we see, okay, it's people who are showing higher amounts of proteinopathy, but also are kind of like advancing in this neurophysiological trajectory, maybe we will have better chances of identifying who's the people at risk. But yeah, I think it's an important limitation, and I don't think that EEG and MEG is going to become the gold standard for diagnosing Alzheimer's disease by itself. I think it can be a great tool to complement other existing biomarkers, but we do have this challenge that given that we expect nonlinear effects across the disease continuum and it's not kind of like a linear change, it's going to be hard to distinguish who's at different stages if we don't have the information of other biomarkers.
Okay, I think that's it for today. Thanks, Jonathan, for coming.
Thank you. Thank you.